Good morning, everyone. Thank you all for joining today's seminar. It is our pleasure to have Dr. Leo Gross here with us today to be able to hear about his work. Leo is a research staff member at IBM Research Zurich and has been there in this position since 2009. He's the team lead of the Atom and Molecule Manipulation Group, and with his team, they work on studying single molecules and on-surface reactions using scanning tunneling microscopy and atomic force microscopy. His group develops and applies AFM, atomic force microscope, as a tool for molecular structure elucidation and investigates single electron charging and transfer amazingly on the atomic scale. His research and work has been recognized by many awards and grants. He's been the recipient of the Gerard Erdl Young Investigator Award, as well as the Feynman Prize for Nanotechnology. He has also received various research grants, including the ERC Consolidator Grant and ERC Synergy Grant. Before I hand it over to Leo to hear more about his fascinating work, I just would like to remind everyone that you can ask your question this time throughout the talk. So please feel free to either type your question through the Q&A option of this webinar, and I can help read out your question throughout the talk or at the end, or feel free to raise your hand. We'll unmute you so that you can directly ask your question. And there's also going to be another Zoom link that Charisse is going to put in the chat box for you. So Leo is going to be available for about an hour after his talk through that link to talk to you if you're interested one-on-one. So if you have more questions or would like to talk to Leo further, please feel free to stop by the other link. With that, Leo, thank you for accepting our invitation and being here today. It's our pleasure to be able to host you and hear about your work. I'm going to hand it over to you.
Great. Thanks for the very nice introduction and welcome to my talk. Thanks for having me at MIT. So you gave a very good summary of what we do already, and I talk about this and in this talk both about on-surface reactions, and then the focus and the longer part will then be on controlling charge transitions by atom manipulation. So playing with single electrons within molecules. And I'll try to give an introduction also that people who are not in the field can understand it as good. And in the end, it will be more and more complicated a little bit. Okay, let me start by introducing the most important people that did the work. So from my team, super important here were Shadi Fataya, who is a postdoc now in my group and was a PhD. And together with Florian Albrecht, he did a lot of the charge manipulation experiments. I'll show in the second part. And Katharina Kaiser, she did a PhD also with us and is now a postdoc. And she did work on cyclocarbon that I will show, so on surface reactions by atom manipulation. So here's the outline of the talk. So I'll introduce you to the techniques. Then I'll talk a little bit of the application to use AFM to identify molecules, and then talk about one example on tip-induced chemistry. And then in the second half, talk about controlling and probing charge state transitions, where I'll show also quite recent work. Okay, so this is a sheet machine. This is the tool we are using. It's an ultra high vacuum scanning tunneling microscope combined with an atomic force microscope. And the sensor of the atomic force microscope is this Q+ sensor. So this is a oscillating prong. Here you can see a magnified image. So we'll oscillate this prong with an oscillation amplitude about 1 angstrom peak to peak. And this is then our tip that we glued to the end of the prong. And we can then detect the tunneling current through that tip, and via the modulation of the oscillation frequency, we can detect the forces acting on the tip. So everything is done at low temperatures, typically 5 Kelvin. Yeah, and that's the most important parts for the introduction. Okay. This is a little summary on STM on molecules. So this is work from Yasha Rep. He did here at IBM before I actually entered the group. And he showed that you can image molecular orbital densities with a scanning tunneling microscope. So, and this is also the model systems we're going to work on. So let's introduce it. So we have a copper or metal substrate. Typically we use copper 111. And then we put a small insulating film on top of it. We use table salt, sodium chloride. And on top of that, we put our molecules. And if we prepare such samples, here it's just 2 monolayers of sodium chloride. Then we have a double barrier tunnel junction. So we can first tunnel through the vacuum into the molecule, and then from the molecule to the metal through the salt. And if we do it this way, we see that we have resonances in the tunneling currents. So this is actually when you attach here holes and at positive voltage electrons to your molecule. And if you now sit on such a voltage and you take an image, then what you observe resembles the density of these orbitals. So something like the wave function squared. And here you see at negative bias, you image the highest occupied molecular orbital. And at positive bias, you image the lowest unoccupied molecular orbital. So sitting here. So here you put an electron into the molecule, and here you put a hole into the molecule. So that's already great. So we can see somewhat orbital densities. Okay, and now we also did AFM. And in AFM, you see something completely different. But first of all, important now to do this kind of high resolution imaging is the end of the tip. Let me come back. Already here, Yasha Rep used a pentacene terminated tip. So often in scanning tunneling microscopy, you make a certain tip by in the end putting a certain atom or molecule at the end of the tip that increases the resolution. And here it was a pentacene molecule at the tip. And now we see for AFM, a carbon monoxide molecule is very good at the end of the tip. So this is then the tip, how you can imagine it. So the carbon monoxide is at the end of the tip, and we make this tip by picking up a carbon monoxide from a surface. So here you have 2 carbon monoxides. We pick this one up, and we see afterwards, the other carbon monoxide changed its contrast. And now if we do this kind of AFM imaging on molecules, we see that before without a CO molecule, just with a metal tip, we basically just see a blob. And now with a CO, we see this molecular structure. In these images, what you actually see is the frequency shift of the resonance frequency of the tuning fork, so the of the Q plus sensor. So basically the detuning, and this corresponds to the derivative, the vertical derivative of the force. But I'll explain this a little bit more in the next slides. Here I want to mention, okay, the important part is this tip functionalization. There's many other tips found by now that also give rise to atomic resolution for AFM. So a few of them are listed here. So noble gas atoms work, chlorine, bromine, iodine, halogen atoms work, nitrogen oxide or oxide, for example. Okay, let's discuss very briefly where this contrast in AFM comes from and what we can measure with it. And here it's nice that you can compare really excellent with theory, because in theory with DFT you calculate energies, and these are easily converted into forces or force derivatives. So we can directly compare our measurements with ab initio calculations. And what we find is that this contrast that you see in the measurements, in a simple explanation, the dark contrast corresponds to attractive forces. So you have like a bathtub shape of attractive forces, and these are mainly electrostatic and van der Waals forces. And then if you go very close with your tip, you see repulsive forces acting in. So these are the bright features, and they stem from Pauli repulsion. And then on top of that, it's important to include the tilting of the CO at the end of the tip. So the CO is not very rigid, but depending on where it is above the molecule, it might be attracted towards a bond or repelled. And this is the reason why these bonds appear so stick-like. And it's also the reason why these hexagon appear so distorted. So you see they're a little bit pulled in the short direction. This is because of the tilting of the CO. Okay, and now the question is what we can use that for besides taking nice images of molecules, which is already also nice sometimes, but Quantitatively, we can determine with a very high accuracy the adsorption geometry of molecules. So that's nice. So here on pentacene, for example, this adsorbs somewhat with a parabolic shape. So the ends tilt up. That's also the reason why in this image, the ends appear brighter than the rest of the molecule. And our resolution here is on the order of a few picometers. Then there's a second thing, and this will become important in the later part of the talk again. We can also distinguish different bonds. And this you can understand once you realize, our contrast is based on Pauli repulsion. Now, bonds and molecules in these aromatic molecules can have different bond order. So they can have like a single bond or a double bond, or of course also something in between. And depending on how much bigger this bond order is, the higher is the electron density at this bond. And this we can see. So if you now look at AFM images, here that's C60, and we have the hexagonal ring on top with two bonds that have different bond order. 3 of them are a little bit stronger and 3 of them are a little bit weaker. And the stronger bonds we see brighter. So this is AFM, where we are in a medium tip height. And we see the stronger bonds appear brighter in this image. And now if you go closer with the tip to your molecule, you see that the stronger bonds appear shorter. They are in reality shorter, but not that much. So this is somewhat magnified by about a factor of 10. And this is again dedicated to the tilting of the CO tips. So I summarize, we can distinguish bonds with different bond order, and stronger, so shorter bonds will appear brighter at a little bit higher tip heights and shorter at closer tip heights. Yeah, and so here this again shows this sharpening effect related to the CO tilting. So here's basically how the CO points at different positions above the C60 towards the C60. And you see here this jump when you cross a bond, and this is the reason for the sharp appearance of the bonds. And right now, these artifacts due to the CO tilting, they are very well understood and they can be nicely simulated here in this pro-particle model from Prokop-Happala, for example. And there's a lot of other works. And of course, one has also to be careful. Sometimes these relaxations of the CO tip also induce artifacts that might look like bonds, although there are no bonds. Okay. So this is a summary of the introduction. This is what you can measure with AFM and STM. I summarize again. So here we have the same molecule, naphthalocyanine, another molecule we like to show atomic resolution and distinguish different methods. With STM, you see the orbital density. So here, for example, the HOMO. And if you do STM with a CO tip, you get somewhat the gradient of the wave function. So you are sensitive to the frontier molecular orbitals. So you select with the voltage whether you want to image the HOMO or the LUMO. Now with AFM, you see somewhat the structure. So now you are sensitive to all occupied orbitals, all electron densities that are occupied add up. And these are usually high above the pi bonds and the atom positions. So you learn something about the geometry of the molecule. And I showed you we can determine adsorption height and also distinguish bond orders. What I didn't show you is there's another mode that is AFM as a function of voltage. This is called Kelvin probe force microscopy. And now with this mode, we again access another property on a molecule. And this is the electric field. And in this molecule here, naphthalocyanine, you see in the center, you have 2 hydrogens that break the 4-fold symmetry. And this we see here in the electric field. So on the arms with the hydrogens, there's a little bit more positive charge, whereas there's a little bit more negative charge on the other arms. And this you can resolve with Kelvin probe force microscopy. Okay, so now we have our toolbox. Let's apply it to do some chemistry and physics and applications.
Leo, if you don't mind, I'll stop you. There's a question.
Oh, yes,
Sharice,
it's good for a
awesome.
question. Good time,
Sharice,
yeah.
can you help unmute?
Yes, I have a question. in your technique at this time, it looked like you can look just atomic interaction between the tip and your probe molecule. Can you look a little bit stronger interaction, such as the interaction of hydrogen bonding between your tip molecule and the probe molecule?
Okay, that's actually a tough question. So there has been quite a debate in the field on the imaging of hydrogen bonds. And it's hard to prove or disprove whether you can see hydrogen bonds or not. In principle, you can form a hydrogen bond, maybe between the tip and the molecule. That should be attractive. No? So here we are actually closer. We reach repulsive interaction even. And there are also works that try to probe this. So yes, in principle, you can probe it, but to detect hydrogen bonds on the surface and whether you would see that kind of contrast, that is expected to be quite weak. And that's, I would say, not 100% solved yet, whether you can see that or not. Okay.
Okay, thank you.
You're welcome. Thanks for the question. So let's continue with the next part. There's a first application that's relatively straightforward. And this would be to identify molecules. So if you have an unknown molecule, you might identify it by taking an AFM image. And actually we did that, and this works. And here we identified a metabolite from the deep sea, hauled up by this submarine. But that's a niche application. Typically, you would use NMR and mass spectrometry as the gold standards for molecular structure identification. And only if you have very few protons, then our technique might be reasonable to use. Okay, but there's another benefit of our technique that we haven't exploited yet. And that's important, and that's its single molecule sensitivity. Of course, in our technique, we image the structure of a single molecule. And that makes it very suited to look at mixtures of molecules. So it's not a pure compound, but molecules are different. Here in this work from the micromy group from Dimas Oteza, they use that to study on surface reactions where you have several products. And we actually then used it to study also natural compounds like asphaltenes. And asphaltenes comprise several 100,000 different molecules. So with averaging techniques, it's very difficult to get individual structures. But now we have the benefit of the scanning probe that we are sensitive to individual molecules. And this is quite an interesting application. And I don't want to go into the detailed findings of these samples. I only want to show you that there are such samples where we can actually use our tool to learn something about these complex mixtures. So these are all mixtures of different molecules, which because of their complexity are hard to study, and where AFM, which is single molecule sensitivity, can give important complementary information to NMR and mass spectrometry studies. So I mentioned already asphaltenes. There are other heavy oil fractions you can study. It's also, we studied combustion products, and here we had quite a good impact on the field, looking into molecules that then form soup particles when they grow. We studied marine dissolved organic carbon, and recently we also studied analogs of the atmosphere of Saturn's moon Titan. Okay. Because of time, let's go into tip-induced chemistry. So this is using the other
Elio,
great
sorry,
thing.
before you go to the next section, there's again a question. Sharice, can you unmute Ali?
I find your lecture very exciting and interesting. We are in the desulfurization, catalytic desulfurization of oil and gas. So my question is, can we use AFM or DFT process for assessing the kinetics of the binding between sulfur molecule and carbon molecule? We're using an oxidation process. Industry uses a hydrogen base, like hydrotreating process. And what we need is very sensitive kinetics testing.
Yeah, so for kinetics, I mean, this is all done at 5 Kelvin. So there is no kinetics here. No? Everything is frozen. And our technique only works at 5 Kelvin. And here we work together with all companies, Shell, Exxon, Chevron, Schlumberger. And the main interest here is in resolving the structures of the molecules. I would not, I mean, you can learn something about the kinetics, but this would be very indirect. So you can use that as input.
I absolutely understand. That's a great explanation. Thank you.
You're welcome. Okay, super. So at the end of each section, we have a question. Good. Let's go to the next section. So yeah, the other great advantage of STM and AFM, and this is what I personally love maybe most about it, is atom manipulation, so that we can do something with a tip. And here we want to do reactions with a tip. And here we actually tackle a long-standing problem that existed in chemistry. And this was the question, how does cyclocarbon look like? Or can you actually make cyclocarbon and stabilize it? So cyclocarbon should be a ring of 18 carbon atoms. And you can imagine to draw two different structures. One that is with double bonds in between all carbons, and one that is with alternating triple and single bonds, which you would call a polyiene, or the one with double bonds you would call a cumulene. Now, the problem with making such a molecule is it's super reactive. I mean, it's very reactive. Maybe you can make it for a very short time, and people have seen glimpses of it, but it immediately will react on in normal conditions. However, now working in our system at 5 Kelvin and on these salt layers, they are important in this context because they are a very inert substrate. The molecule doesn't form bonds to it. We can try to make such molecules from precursors, and then we can study them. So this is the work Katharina Kaiser did the experiments, and it's a collaboration with Harry Anderson groups from Oxford. And they made this precursors, so Harry Anderson and Loris Scriven. And here the idea is that now with pulses from the tip, you take off these CO groups, and you typically take them off pairwise. So you take off two COs from one side with a voltage pulse, and starting with this molecule, and here you see its AFM image on the salt layer on the surface, we take off these two carbon monoxide and we end up with this molecule. And we do this by voltage pulses on the order of 3 volts. And we can take off another pair of COs and yet another pair of COs, and then we actually end with a ring. So this is cyclocarbon. And now in our conditions, it is actually stable on this double layer of sodium chloride, and we can study it. And already from the image, you might have a good guess what's the correct structure. The image shows 9 lobes, so it's a 9-fold symmetric structure. So actually it is a polyionic structure that we find on the surface. And we can also, if we go now closer with the tip, and remember this related to our first slide where we looked at bond order, where we see that bonds of larger strengths appear brighter. So we see now bright these triple bonds, each one as a lobe. If we go closer, then the CO tilting starts to become important. And now we actually see corners at the position of the triple bonds. And we can simulate this with this probe particle model from Prokop Happala. And we see that if we put in the structure of a polyionic, which we get from calculating DFT with one code, we get a very good agreement. Whereas if we put the cumulonic structure, which another code delivers, we get a very much symmetric structure that looks like a ring. And from calculations, it was not possible to solve which one is a ground state. But here we see now really on our surface, at least for our surface, we can answer that questions, and it's a polyionic structure that is formed. Also interesting about this molecule is one might use this as building blocks to further construct nanostructures on surfaces. So here we have 2 of these precursors. So some of the CO masking groups have been detached. And now with voltage pulses, it's sometimes possible to fuse these precursors. And so that would be interesting to form larger carbon-rich structures on surfaces. And recently, we also used another precursor, and we could increase the yield to form cyclocarbon on the surface by atom manipulation. All right, this already concludes this tip-induced chemistry part. I only wanted to show this one example. And in the last part of the talk, so for the next 15-20 minutes, I'm going to talk about controlling and probing charge state transitions. So now we will play with single electrons in addition. This is the introductory slide. And here you show already previous work that has been done with single charging. So this is work from Yasha Rep again, where he showed that you can charge single gold atoms on a salt layer. So this one is neutral, you can charge it negative and you charge it neutral again. And here you see in STM, you see that typical dark halo for the negative charge state. You can also distinguish these charge states in AFM. This is again using Kelvin probe force spectroscopy. And here you sit with your tip on the molecule and you record the frequency shift as a function of bias, and then you get a parabola. And the peak of this parabola tells you something about the charge. And important is the lateral shift of the peak. And always when the peak shifts to a higher voltage, that indicates negative charging. So here you see the peak shift going from a neutral to a negative gold atom. And the blue parabola therefore is shifted towards positive bias. Okay. Here was the previous work where a single molecule was charged on a bilayer, but on a bilayer, this only works in certain cases when you have charge bistability, as for these gold atoms and these molecules here. Now, what we actually want to do is we want to go on a very thick film. And then when we put the molecules on a thick film, let's say thicker than 12 monolayers, then we can charge them with electrons and they cannot tunnel to the substrate. The only channel to decharge them is either going to another molecule on the surface or going back to the tip with a charge. Okay, and here I show you how this works. So we go with the tip above a molecule and then we record this parabolas again and the frequency shift. So we cannot do STM anymore. We're on a thick film now. So there's no tunneling current, just single charges that we can attach from the tip to the molecule or back. And here we see in this parabola, we get then a jump when a charge tunnels from one parabola to another. So here we charge a molecule positive, and when we go back, we might charge it neutral again. And on the other side, we might charge it negative. And this we can do in a controlled way. So here are 4 pentacene on a thick film, and we go with the tip above one, we charge it negative, we go above another one, charge it negative, charge this one positive, and we take images in between. And because we land on these different parabolas, you see them here with different frequency shift. So this shows we can control the charges of molecules on these thick films. What we want to do next is now we want to combine this high resolution imaging I showed you in the first part with this controlled charging that I showed you in the second part. So what we do now is we first make a CO terminated tip, so that we have this tip that gives high resolution. Then we actually go to the molecule that we want to image on a thick film. And now we look at this parabola. So we identify its different charge states. So here on pentacene, we can actually see 4 charge states: positive, neutral, negative, doubly negative. And now we sit on such a voltage and go close with our CO tip and take an image. Now, either at this voltage, for example, the image will be of the neutral molecule, but if we take it here at plus 2.5, it will be of the negatively charged molecule. And this is what you get. So this is now pentacene, and so these images are acquired at the voltages corresponding to the different charge states. So positive, neutral, negative, doubly negative. Now it's not so easy to compare directly bond lengths in each images because the different voltages will also lead to different distortions due to the CO tilting. But you can compare qualitative differences within the images with respect to each other. So for example, here, what you see is that for the negative and the doubly negative, ring number 2 and ring number 4 become darker. Yeah, and also here you see it. And this, for example, you can rationalize with this Kekulé drawings. Now, if you have the neutral molecule, you would draw it like this, and you can shift this one bond at different positions in this Kekulé representation. Now, if you charge it doubly negative, you really have this structure that you would like to draw as a chemist, because now you have 3 Clark sextets, and you put these additional charges in ring number 2 and 4, which therefore increase this bond length. And this is actually what you see in the image. So we see actually now changes in bond order related to different charge states. And we did a couple of molecules. I only show the second one here because I like it a lot. It's porphine, so it's a parent compound of hemoglobin or chlorophyll, with these charge states being very important for life, of course. And in porphine, you can have this conjugation pathway in the neutral molecule. No? So this is where you alternate your bonds in these 2 resonance structures. And here we see the neutral molecule, again imaged with AFM. Now let's compare the neutral molecule with a doubly negative molecule. No? So when we measure here at 3-point-something volts, 3.2. If you draw the structures, you see that now the conjugation pathway probably would change. No? So you can now draw the whole outer periphery of the molecule. And let's focus at these 2 bonds on the rings without hydrogen. And you see that for the neutral charge state, they were double bonds in both resonance structures. And in fact, this bond appears short here. Yeah? And now if you look at the negative charged one, you see there can be single bond in the one representation or double bond in the other. And in fact, if you now look, you see we have a much longer bond here. So longer bond appearance for for weaker bonds. Exactly what we see. And there's also other fine differences. For example, you see a larger bond length alternation in these bridges here, which would indicate that in B, this resonance structure is the dominant one and this the less dominant one. Okay, so you can see different structural changes upon charging with this technique. Okay, now for something different, again with charging, but now we are more interested in the energetic landscape. So we want to do spectroscopy. I showed you this image before. This is what you can do with STM on a double layer. Now you can have a neutral molecule and you can charge it negative or positive. What you cannot probe is way back. So once you charge it negative, how does it look like when it now becomes neutral again? So the discharging, because this you cannot control, it happens by this tunneling through the substrate. Now in our method now on the thick film where we change electrons back and forth, we can now probe both directions actually. So we can probe the charging and the discharging. And this is what we want to do here. And in this Kelvin probe, force microscopy parabolas, this would be this hysteresis here. Okay, and that's actually interesting to probe that because what you get out of that is the reorganization energy. So if you charge a molecule in this Frank Condon picture, you go from one parabola to another parabola, but the charge transition is fast. And then the structural relaxation is slow. And this is the relaxation energy that you then gain during the structural relaxation. And what we want to determine here is then the sum of these 2 relaxation energies, charging, discharging, and the sum of these 2 lambdas is the reorganization energy. And it's a very important parameter for electron transference systems. And this we gain exactly if we compare the charging and discharging energies. How is the time? Oh, we're far already, so I'll keep this rather short. This is here a little bit the detail of the experiment, but it's actually quite nice to look at it. So what we do is we first charge the molecule to a known charge state, let's say plus in this case, and then we retract the tip a little bit so that the tunneling probability becomes so low that we have an event in maybe once in a second or once in 10 seconds or once in 100 seconds. So slow enough that we can actually measure it with the AFM and see when the force changes because the molecule has been discharged. So here we have 10 seconds, we charge the molecule, and after 4 seconds, the molecule has been discharged. Of course, that's now a single experiment, and this is stochastic. It's an exponential function. So we have to do it several times to get the lifetime from averaging. So we do it 80 times, and then we get a a histogram of how long it takes to discharge the molecule, and we can access the lifetime. We do it 80 times now for each different voltage. And then we get basically this curve, which gives us the ratio of this peak as a function of voltage. And from this, we can then determine the lifetime. And now the nice thing about it is we can do it for both directions. So this graph shows the charging going from neutral to positive, what we could have measured with STM on a bilayer film already. And you see how this lifetime changes. If we take the derivative, we actually get something like a scanning tunneling spectroscopy spectrum, and we get the energy at which we charge the molecule positive. Now, only with AFM, we can measure the reverse progress. So going the discharging from positive to neutral. And with the same method, we also get here the curve. And now here we get the energy at which we discharge the molecule into the tip from positive to neutral again. And the difference of the two corresponds then to the reorganization energy after taking into account for some corrections. Okay, and here's the result of that. The reorganization energy that we determine in this case is 0.8 electron volt. And this is a high value. And this high value is understood because our molecule is on salt. And the ions of the salt, they relax to the charge. So the most of this energy actually goes into displacing the ions of the salt film. And we did DFT calculations, and it's actually in a nice agreement with the experimental value that we found. A side remark, so this is not work of our group, but work of the group of Yasha Rep, done by Laerte Patera as first co-author. And he automated this method so that you can take now images for this charging and discharging. And this is done by syncing voltage pulses to the oscillation of your cantilever. And here, for example, you see how you put an electron into the LUMO of this molecule, and you see it's 4-foot symmetric, but that's because you can put it, these two orbitals are degenerate in the neutral charge state, and you basically see their sum. But if you now empty that orbital again, now what is not easily done in the STM measurement, but has to be done here with AFM, you see only one of the two orbitals because of the Jantella distortion that actually prefers one of these two states. Okay, let's go one step further. Can we create and probe excited states? So we do the same thing as before, but in the discharging, we don't want to go back to the neutral molecule in the ground state, but in an excited state. So this is the experiment. So this is what we did before, what I already showed you in this reorganization. Now I don't show you the charging, it's the same thing. We charge it positive again. And now we probe the discharging. We can either do it here, or if we go further away to limit the tunneling probability, we see that another discharging channel opens at larger voltages. And if we yet go further away, another third discharging channel opens at larger voltages still. So we have two other reduction channels that open. And here is the picture how we explain this. So again, we can convert this to lifetime and STS spectra and extract energies. And basically now we get an energy for forming the molecule in the neutral ground state. So that would be going to positively charged and back to S0. Or the next higher energy should correspond to the first triplet excited states, and the next one to the first singlet excited states. So here the green arrow. And so then we can actually determine these different energies. So for the single molecule, the excitation energy to the triplet and singlet excited state. We compared this with literature. In a nutshell, our experiment is lacking a little bit accuracy coming from this. You have to do all these measurements with single electrons, so you have quite a large error. But the values that we get, they are feasible. And here, the nice thing is we can do that for individual molecules and also on an insulating film. Okay, and what's also nice in these measurements is that we can see actually which state we occupy, whether we go into the neutral ground state, triplet excited or singlet excited state. and we can compare the different branching ratios into these different states, which would correspond to the ratio of these colors here. So at this voltage, we would form 2/3 in the triplet excited state and 1/3 in the neutral ground state. Okay. Final thing, just another 2-3 minutes. I want to show this work. This is done in the group of Yasha Rep, mainly by Yingbo Peng. And I was grateful to collaborate here. And here now they automated the second thing, but in a very cool fashion. So you, again, the molecule, this experiment starts similar. You charge a molecule positive. Then you go back to the neutral state, either in the triplet or singlet. And then a third pulse now actually maps this excited state into the charge state again. And you will only end up in the charge state if you were in the triplet state. Now you can vary your dwell time. So this time here. So when you like the time from the second to the third pulse, when you probe your system. And with this, you can then actually probe the lifetime of these triplet excited states. And this is shown here for pentacene. And they showed that actually you don't get a single exponential decay, but a much better fit to a triple exponential decay, which makes perfect sense for a triplet ground state. And they could determine these 3 decay constants, which fit exactly to other measurements that had been done on single pentacene molecules already. So these are 2 papers from '93, published back to back in Nature. And the decay rates are very comparable. And these were already measurements on single pentacene molecules in a host matrix. But now the super cool thing is that here Yasha and Yingbo, they can of course also have the access and take atomic resolved images now of the single pentacene molecule and its surrounding. And what they did here is that they put single oxygen molecules, O2 molecules, next to the pentacene. And now you can observe how the lifetime of the triplet state quenches depending on how many O2 molecules you put on around your pentacene molecule and at which atomic positions exactly you put these O2 molecules. And this all makes a huge difference. And of course, this could not be measured before and can rise to a much better understanding of this quite important triplet quenching in molecules. Okay, with this, I want to summarize. So in the last part on this charge manipulation, I showed you that we can control and detect charges on molecules. We can obtain atomic resolution and bond order analysis of molecules in different charge states. Here, see again in this movie, the porphine molecule in its 3 different charge states. And it's not running anymore. Never mind. And I showed you that we can determine the reorganization energy with AFM. We can determine energies of excited states. And then in this work from Yasha Reb, I showed you, and Jingbo Pen, I showed you the triplet lifetime measurements. This brings me to the acknowledgments. So Shadi Fataya is this guy. He was the most important person for these charge manipulation experiments that he did together with Flo. And Katharina, she was most important for the C18, the cyclocarbon paper that she did together with Fabian Schulz. And I'm very happy for my former group members and for Yasha Rep. I showed a lot of his results to Franz Giesebel, on whom we collaborate with in terms of the Q+ sensor development. Mats Persson for theory, and Diego Peña, Harry Anderson, and David Fawkes were chemists that synthesized precursor molecules. I'm thankful for funding. This ERC Synergy grant, where I continue this research that I showed you with Yasha Rep and with Diego Peña, just started. And here I'm looking for PhD students, master students, postdocs. So if you're interested, please apply. And yeah, thank you for listening to my talk. And now I'm ready for more questions.
Thank you, Leo, for a fascinating talk. So we have time for a few questions. Again, feel free to raise your hand. We'll unmute you. You can ask your question directly or type your question in the Q&A box. Okay, so there are a couple of questions already written. One question is asking, in C60, each carbon-carbon bond is 50% a single bond and 50% a double bond. How could you resolve the single and double order bond here?
Okay, let me go back and clarify first. So in C60, the two different kinds of bonds, they are not single and double bonds. So the difference is much smaller in C60 than between a single and a double bond. But I'm sorry, this is a bit awkward. Should have done that differently. Okay. Here we are. Yeah. So actually, I forgot how many different representation of C60 you can make. But if you say this is a double bond with bond order 1, and this is a single bond has bond order 1, and the double bond has bond order 2, in this representation, then the bond between hexagons and pentagons, they would be bond order 1.28, and the bond between two hexagons, there would be 1.44. So that's a little bit stronger, no? But it's not like 1 to 2. And if you compare the real difference in bond length, if I'm not mistaken, right out of my head, it's a few picometer, and that's we would not be able to resolve that. So how do we actually see the difference? So one thing is they differ in electron density. That's this brightness contrast. So why we see this with different brightness, these bonds that are stronger, the ones between two hexagons. And now if we go closer, then the CO tilting leads to this shortening and lengthening of bond, also because larger electron density will lead to larger repulsion, which will also lead to a larger deflection of your CO. No? So the tilting will increase. That's why it likes to basically avoid the positions with high electron density. That's why these corners in C18, the triple bonds become corners. And here these double bonds, or not double bonds, shorter H bonds, and because of their larger electron density, become so much shorter. So this is how we can distinguish them. So it's not fully quantitative, no? We can say this bond is smaller and this is weaker. And also it's kind of difficult. The molecule needs to be very planar to do that, because if you have height differences, this will also change the tilting. But then we can semi-quantitatively measure or distinguish these bond length.
This is Juan Herbsommer from Texas Instruments here in the US. So really impressive presentation. Thank you very much. So I guess that my question is rather simple. I will say I was thinking about the 3D nature of the molecules you know. So I was wondering, you don't do a conformal movement of the tip over the molecule, correct? So I think that you keep a z distance constant, correct? So in the case that you have planar molecules, okay, that I think that's fine, okay. But I would say my question is, what is the importance of, let's say you have a molecule that is really 3D nature, okay? It's not planar. How do you account for that, okay? Or how do your technique actually deals with that, or how the distance affect the results, okay?
Yeah, excellent question, of course. So there's two answers. The first one is, okay, one thing that we can do is we can follow a little bit the topography of the molecule. Okay. No? So in this case of C60, we could see parts of these next rings that surround the top tile. You're right. So this image is taken just as constant height, and that's why we just see the top hexagon. We can follow with the tip a little bit the C60, and sometimes we do that. You can use AFM feedback or STM feedback, maybe sometimes. Yeah, for example, here we did it on this molecule. So this image, you resolve a little bit more because you follow the topography a little bit better. Okay. On the other hand, then it's already harder to interpret the image because now you have you fold in different types of contrast. Okay, but also with that, you just probe the first atomic layer of the molecule. So our technique with this CO tilting in AFM is super surface sensitive, which is nice because we see atoms and bonds and all these great things. But it's really hard with this technique to look inside of molecules.
Okay.
So that's going to be really tough. So, and there I don't really, I mean, you need something else or something else in addition to be able to look inside a molecule. So our technique is really good for planar molecules. It can still be adopted to a little bit tilted or non-planar molecules if they are in one plane, let's say.
Okay.
But large 3D molecules don't work so well. And so, but again, there are a lot of planar molecules which are interesting, and many samples, there are the planar molecules that are the most interesting stuff, like in these, no, for example, heavy oil fractions or combustion or.
No, that's perfect. That's
Yeah.
I think you answered my question. I think I'm not questioning the technique. I think it's super awesome. You know, I'm amazed that you guys can do that. Okay, thank you.
Thank you.
Great. So Kate is asking, how do you ensure that the molecule is lying flat on the substrate?
Yeah, so we can't do much about it. Typically, molecules lie flat. Yeah, and so because they want to maximize their absorption energy, you know, and typically all these flat molecules, they like to be planar. There are some exceptions, though. I think I didn't have it in this talk, and I'm not sure. I probably also don't have it in extra slides. But sometimes you can actually change that. So you might flip a molecule up or down. So we had one molecule where we could actually, when we charge it neutral, it would stand up. And when we charge it negative, it would lay down. So there are some special cases where you can actually change that. But typically, most molecules are planar and we can't really change it. Sometimes you might change it with charge manipulation.
Great. So the next question is asking, could you tell us more about the method to put oxygen at different positions for triplet state measurement?
Yeah. Okay. That was not my work. So that was work by Jingbo Peng in Yasha Rep's group and Yasha Rep. And what they did is they dosed oxygen, and then basically with voltage pulses, you can like sometimes move it. I don't think it's super controlled, but here you just want to change the position. No? And I think in this case, you would never see oxygen on the free surface, but it would always absorb next to something. It's like here to a pentacene molecule. And then you can see it, you can have it in different places, and you can move it a little bit or take it away or bring other oxygens close by. And I mean, typically in all these methods, you have your tip, no? And then in general, for atom manipulation, you have the two possibilities to apply voltage pulses with the tip. And then something might jump away or under the tip, or a reaction might occur, as we've seen in C18. Or you can also go very close with the tip and then try to pull or drag adsorbates across the surface. On salt, typically these voltage pulses work much better, and this pulling and pushing doesn't work so well because you pick up your adsorbates if you do that on salt, because they like the metal tip better than the salt where they are on.
Thank you. So the next question is asking, the techniques you talked about, do they work for measuring band gaps or work functions of semiconductor doping or graphene doping?
Yeah, so in principle, you can measure work functions and band gaps with these methods. So we didn't work on these substrates. We worked a little bit. Okay, so we did some work. And yeah, so there's different methods. So Kelvin probe force microscopy is actually very suited to do that, to measure work function differences. And also you do that by scanning tunneling microscopy if it's possible, by looking at the decay of the tunneling current as a function of distance. You can also determine work functions. Yeah.
Great. How about biomolecules? Would you be able to measure DNA or linear proteins?
Yeah, that would be nice. There, the challenge is twofold. One is the preparation. No, so the molecules that I showed you, typically are thermally evaporated, which means they can't be very huge. So with long DNA strength, this wouldn't work. No? This molecule would break before you sublime it into the gas phase. So there's this first challenge of preparing it. And then the second challenge is again imaging, because they might be a little bit non-planar. But in principle, yes. And actually there's work of the group of Ernst Mayr that imaged DNA using exactly this technique. The problem is a little bit then this DNA comes on the surface and it forms a 3D molecule. And now again, you can't look inside, as discussed before. So you see some of the atoms on the outer part of the molecule. So ideally, you would prepare such a DNA as one string that is nicely aligned, and then it's more or less planar, and then you could take a high-resolution image. This would be great. Preparation is very difficult then.
Makes sense. So we're out of time. Thank you very much, Leo, for an amazing talk. We enjoyed it very much. Thank you for taking the time to be here. Thank you to our audience too for joining us today. The next